So when we talk about fermentation reactions in yeast or in bacteria, all of this is due to enzymes. So it's enzymes that's converting the large macromolecules into small molecules. And none of these reactions could happen if they were just sitting around at room temperature. The same is true in our bodies. When we eat food and we break down the large food macromolecules into small ones and we get energy in the process, this could not happen at room temperature or the temperature of our bodies. And so we need enzymes to speed up this process. Enzymes are proteins, which means that a lot of what we already know and have learned about proteins is also true for enzymes. But enzymes are very special proteins, and we're going to learn more about that in this segment. So, for example, some of you may be lactose intolerant. And what this means is that you have a lack or a deficiency in the enzyme that converts lactose to its energy sources and breaks it down. And this protein is called lactase. And in fact, many enzymes end in the ending ase, so lactase, protease, peptidase, and so on. So when you are lactose intolerant, you can take a pill that contains lactase, and this will then, if you take this just before eating a dairy product, this will help you break down the lactose, and you've basically taken care of the problem artificially. So what enzymes do is they make chemical reactions happen faster than they otherwise would. So if sucrose were just sitting around at room temperature, it may eventually break down to fructose and glucose, but in order to speed this up, there are enzymes that basically cut those bonds and make it happen faster. Enzymes speed up reactions either by helping to break bonds or by helping to make bonds happen and make bonds form. And in both cases, enzymes often work by binding to the molecule or molecules in question. And in terms of the bond breaking, they will then facilitate this bond breaking happening faster by rearranging the molecule or by affecting the atoms in the molecules in some way. In terms of making bonds form, enzymes do this in a simplified way by bonding to two molecules and kind of bringing them closer in proximity. And this bond can then form more easily because the molecules are positioned in a way that make the bond happen more easily. And so you can understand from this then that the enzymes need to be very specially designed to fit the particular molecules they work on. And so for each type of chemical reaction that an enzyme catalyzes, it is designed to kind of help that reaction. And so it is the three-dimensional structure of the enzyme that is important for this, for binding to the molecules and for causing the reaction to happen. And so if this three-dimensional structure is broken or messed up in some way, you can imagine that the enzymatic reaction would not happen. Another way to think about this is in terms of activation energy. So the energy that is needed to either bring molecules together and make a bond form, or the energy that's needed to make the bond break, that is the energy we need to put in to make the reaction happen. And this is the activation energy or the activation barrier. In the review materials of this course, we talked about how you can basically write out a chemical reaction on a diagram where you show that the reactants go to products, and we showed you how there's kind of this hill in energy, and so the energy needed to bring the reactants to the products needs an input of energy, and this is the activation energy or the activation barrier. And so enzymes work by basically lowering this barrier, and so you can imagine then that the products would form more easily and the rate of the reaction would increase. Enzymes are important for food and for cooking in many ways, and in fact in many ways that we may not think of normally. So for example, when making cheese, the enzyme renin is what's causing the casein molecules to lose their negative charge, which makes them fall apart, and this causes the coagulation of the casein, which then leads to cheese forming. Similarly, when fruit or vegetables brown, this is also due to enzymes. So what happens when you bite into or when you slice into fruit or vegetables is that these enzymes that were in special compartments of the cell are then released, and they can react with other compounds in the fruit or vegetable. So America's Test Kitchen will tell you in their video about how to make pesto. They will tell you about how this happens when we slice garlic and the flavor molecules are released as a result, or when we cut up onion and this leads to reactions that causes very strong compounds that causes our eyes to tear. In the case of browning fruit, the enzyme that causes the browning is in a separate place compared to phenolic compounds. And when you bite into a fruit or you bite into an apple, what happens is that these enzymes react with the phenolic compounds. And originally, the phenolic compounds were supposed to react with attacking insects or animals, but the browning enzymes then react with these compounds, and this causes, together with oxygen, these large brown compounds. And this is what we see when we let an apple or a banana or avocado just sitting out on a counter for some time. Since enzymes are proteins, they're going to react in many ways just the same way that proteins do. So for example, when we apply heat to enzymes, they're going to denature and recoagulate, just like the proteins did when we talked about cooking meat in the heat transfer week. Similarly, when we discussed ceviche and adding vinegar or lemon juice to fish, and this caused the proteins to denature, this is a similar way that enzymes will react if we lower the pH and hence change their internal structure in some way. This is actually something we can use when cooking. So for example, the enzymes bromelain and papain are enzymes from pineapple and papaya. And these are often found in meat tenderizers because they break down the proteins and so thus makes the meat more tender. Now, if you've ever tried to cook pineapple jelly, the recipe probably told you to heat the pineapple in some way before adding the gelatin. And this is because if you don't do this, the bromelain enzyme will actually break down the gelatin, and so the gelatin will not form the jelly that we want when making the fruit jelly. Similarly, America's Test Kitchen will show you that if you add an acid, lemon juice or ascorbic acid when making pesto, this will keep the green color of the pesto rather than have it turn black or dark like it otherwise would. And this is also why you would add lemon juice to avocado when it's just sitting out before adding it to a salad. You then inactivate these enzymes. The lower pH messes enough with the enzymes so that they will not cause these brown compounds, and thus the avocado stays green. What's true for all chemical reactions, regardless of whether they're helped by enzymes or not, is that a small increase in temperature, not so much that the enzymes are denatured, but a small increase in temperature will usually speed up the reaction, and sometimes even a large increase in temperature will do this. And you know this intuitively from baking bread. You know that you put the dough to rise at a somewhat warmer temperature, or you know that if you want avocados or bananas to ripen, you leave them outside of the fridge. Collagen is another example where Nathan told you that if you slow cook meat, so you keep the temperature not too high, but at a temperature around 50-60 degrees for a long time, then this is enough to slowly break down the collagen over time. And this would take a very, very long time if you were to just do it at room temperature, but that small increase in temperature is enough to make the reaction happen faster. So temperature increases the reaction rates. Another way to think of this in terms of our reaction diagram is that an increase in temperature somehow manages to make the reactant molecules turn into products more easily. And so this increases the likelihood of a reaction to happen. So if you think about this, if you wanted two molecules to react with each other, they needed to you needed to increase the likelihood that they would be in a position so that they could react. And one way to do this would be to just make them move around more, and then the likelihood of them bumping into each other and causing a reaction would increase. So how would we do this? We would increase the temperature and then they would move around more. All of this is described in what's called the Arrhenius equation. Arrhenius basically described the effect that temperature has on the rate of a reaction. And here is this equation. So the Arrhenius equation says that K, which is the rate of the equation, equals K0 times e to the negative delta U over KT. So K is the rate of the reaction, and K0 is the maximum rate that each molecule could have if it had the maximum energy needed to kind of get over that hump or get over that activation barrier in the chemical reaction. Delta U is that energy barrier. So the higher the barrier is, the more energy we need, the higher delta U is. And kT, of course, we know from before, that's just the thermal energy. And so the higher the thermal energy, the more the molecules move around, the more energy there is for this reaction to happen. So what this equation tells us is then that if the temperature, if the thermal energy is very large, then the exponent becomes very small, so it becomes very close to 0. And e to 0, of course, is 1. So this means then that the rate would be very similar to k0, which is the maximum rate. So high temperature would mean a very high rate, very close to the maximum rate. Similarly, if we have a very low temperature, then the exponent would be very large. And this then would give us a reaction rate that is very different, much smaller than the maximum reaction rate. And along the same lines, if we had a very big delta U, we had a very big activation energy, then the exponent would be large, and this would then cause a reaction rate that was also lower, much smaller than the maximum reaction rate. So this equation may look complicated, but it's really just building on what you already know. You already know about thermal energy, and it's telling us that there is some maximum rate that a chemical reaction can have, and that the actual rate then depends on an energy that you need to put into a reaction and how much thermal energy you have around. So in the case of collagen, for example, and slowly heating the collagen at low temperatures, what happens is that the collagen fibers, which is this long string, it's a triple helix, will break down into gelatin. And there are several steps to this, but one of them is hydrolysis of the peptide bond. And so you can imagine as you slowly increase the temperature, the likelihood of this peptide bond to hydrolyze and break down into gelatin increases because the rate of the reaction increases. And this is what happens when you break down collagen or in many other chemical reactions in cooking.